Okay, this section of notes is going to be on RNA. So we learned about DNA and how DNA is built and the function of DNA. So in your cells, they want to keep the DNA safe. So DNA is going to stay in the nucleus, but it's got to have some way of communicating its content and message out to the rest of the cell. It does this in the form of RNA. So we're going to start with that. So one thing that you should get out of this by the end is understanding how RNA and DNA are different in their structure and then also how they function. Okay, so what is RNA? On the right, you've got a picture of an RNA molecule. You're going to notice something right off the bat. It's single-stranded. So one single strand. It's made up of a sugar, a nitrogen base, and a phosphate group. So just like DNA is, that sugar is ribose sugar though. So that's how it gets the name RNA instead of DNA. The four bases, so if you remember from DNA, you have A, G, C, and T. In RNA, you're going to have A, G, C, and U. So in our picture over here, this brown uracil is the different base pair that's in RNA. And still going along with the theme of this slide, what is RNA? RNA basically puts the genetic code into action. So if DNA is like a storage of your genes and what makes you you, RNA starts to put that recipe into action. So now let's look at the differences between RNA and DNA. Three main differences. The first one is that the sugar is RNA. Sugar in RNA is ribose, not deoxyribose. Second is that an RNA is generally single-stranded. So here in your visual comparison, you got your single strand here versus your double helix of the DNA. RNA contains uracil instead of thymine. So you have the sugar backbone is different, single strand, and that single base pair that's different. And it's uracil instead of thymine. So what is the function of RNA? We talked about RNA putting the genetic code into action, and how does it do that? So if we look at our little diagram over here, we got the guy looking at the master plan. Master plan is like the DNA. That's the original source of the information. But let's say you have this message and you need to get it out to a bunch of workers in an area. What you're going to do, instead of taking this original master copy, which might get damaged if you sent it out into the field to get worked on, you're going to make a bunch of copies of it. And you can send those copies off to several different workers, and they can get started on different functions in your workspace. So in the cell, you've got your DNA. DNA is copied. Those copies are sent out. They're going to be sent to ribosomes. And what happens at ribosomes? We make proteins. So that's the most important function of RNA, is to make proteins. Okay, three types of RNA. So they're basically like three different types of workers in the cell. Messenger RNA is going to be the thing that's going to start in the nucleus. It's going to get, it's a copy of the DNA. So the messenger RNA gets copied from the DNA. It's going to get tagged and sent out of the nucleus of the cell. And it's going to get carried to a ribosome. The message in that RNA is what's going to make the protein. It's got the message or the instructions for what to make. Then you have ribosomal RNA. So an RNA is basically made up of two parts, and in here they've given you like the blue and the gold or yellowish part here. RNA is built out of, or a ribosome is built out of RNA. And the RNA in the ribosome basically helps to read and attach this messenger RNA. So the messenger RNA is going to slip right in between these two things here. And the RNA in this ribosome is going to help read it. And then the very last one is this transfer RNA. So this is the third type of RNA. And They take the amino acid here, um, and they're going to read the code. They're going to match up with the code in messenger RNA, and they're going to attach amino acids. So they're going to transfer amino acids to the ribosome. And we'd have several transfer RNAs coming in. They would keep coming in, and they would read along the length of the messenger RNA. Each one would attach, and as it came in, it would attach an amino acid. And the string of amino acids together is your newly formed protein. So that's the function of the transfer RNAs, bringing those amino acids in. That's the end for this slide. We're going to break this into several small steps. Each process is complicated, so I want to focus on this one just on understanding what RNA is, how it's different than DNA, and the three types of RNA. Review those and understand them well, and then we'll talk in more detail on how they function within the cell.